In vitro transcription of RNA. Required consumables and equipment: PCR tube, PCR plate, vortexer, pipette, icebox, centrifuge, constant temperature drying box, PCR machine. Reagents required: HIFER T7 high-yield RNA synthesis kits, 10623. To improve RNA quality, it is recommended to use plasmid linearized templates for transcription. First, briefly centrifuge T7 RNA polymerase mix and place it on ice. Next, thaw 10 times transcription buffer and ribonucleosides from T7 high-yield RNA synthesis kit, and vortex them to mix well, followed by centrifugation, then place them on ice. To avoid RNase contamination, wear gloves and thoroughly clean bench surfaces, pipettes, and other materials. To synthesize RNA, reaction should be carried out at room temperature, which is added 10x buffer, nucleotides, template DNA, RNA polymerase accordingly. The order in which reagents are added is critical for RNA transcription. The above reaction solution should be mixed well and centrifuged briefly to the bottom of the tube, and incubated in a dry air incubator for 2 hours at 37 degrees Celsius. Product purification. Required consumables and equipment: PCR tube, PCR plate, vortexer, pipette, icebox, magnetic stand. RNA can be purified using the HEF-NGS RNA Cleaner Purification Kit. First, remove RNA clean beads from 4 degrees Celsius and equilibrate them to room temperature. Dilute the transcript with RNase-free H2O. Vortex to mix the magnetic beads thoroughly. And then pipette 2 times magnetic beads, 100 ml, into the RNA sample, 50 ml. Mix well by pipetting several times. Incubate at room temperature for 5 minutes to allow RNA to bind to the beads. Place on a magnetic stand for 5 minutes. After the solution is clear, carefully remove the supernatant. Add 200 ml of 80% ethanol freshly prepared with RNase-free H2O to rinse the beads. Incubate for 30 seconds at room temperature. After that, carefully remove the supernatant and repeat this operation once. Air dry the magnetic beads with the lid open for about 5 minutes, avoiding over-drying. After fully drying, add 22 ml of RNase-free H2O, and mix by pipetting several times. Let stand for 5 minutes at room temperature. After the solution is clear, carefully transfer 20 ml supernatant to a new RNase-free PCR tube. RNA size and quality detection. Required consumables: instruments, nanodrop spectrophotometer, Agilent 2100. Reagents required: samples purified as above. Using UV absorption, we can determine RNA yield by measuring the UV absorbance of the product at 260 nanometers. The Agilent 2100 bioanalyzer can be used to assess the integrity and quality of RNA. It requires only a small amount of RNA for analysis, and high-quality RNA should show distinct and sharp peaks on the electrogram. HIFAIR T7 High-Yield RNA Synthesis Kit 10623 can synthesize long transcripts and short transcripts, and can generate 100 to 200 μg RNA with 1 μg template input. Synthetic RNAs can be used for downstream applications such as RNA structure and function studies, RNase protection, probe hybridization, RNAi, microinjection, and in vitro translation. RNA co-capping. Required consumables and equipment: PCR tube, PCR plate, vortexer, pipette, icebox, constant temperature drying box, PCR machine. Required reagents: 10614 kit, 10612 kit, 10619 kit, 10132 kit. For the standard capping reaction, first dilute 10 μg of purified uncapped RNA to 12.5 μl with RNase-free H2O, and heat the RNA at 65 degrees Celsius for 5 minutes to disrupt its secondary structure. Then immediately place on ice for 5 minutes. Begin the capping reaction by sequentially adding capping buffer, GTP, freshly diluted S-adenosylmethionine, SAM for short, vaccinia capping enzyme, and mRNA cap2-oxymethyltransferase to the denatured RNA. The above reaction solution should be mixed uniformly, briefly centrifuged to the bottom of the tube, and incubated for 1h in a dry air incubator at 37 degrees Celsius. At this point, RNA capping with Cap1 is completed, and subsequent experiments can be performed. After incubation, the RNA is purified and analyzed according to the method of product purification in the previous section.